So Gayatri, when was the last time you were in the campus?
I am trying to remember, maybe about 4 years ago, I think. Yeah.
So you didn't come to the new campus?
No, we actually, yeah, in fact, I stayed in the guest house. Last time I came, we actually stayed in the guest house, but labs were still in the other place.
Okay.
From what I remember, that is what it was. Yeah.
Right. So this COVID actually, completely messed up the plan. I was
Sorry to interrupt, sir. We are live now.
All right.
So, Anandita, you can start the program sharp at 4 o'clock.
Another 2 minutes.
So we are live now.
2 minutes to go.
Okay.
I think Vikram.
Hey, Moli. Sound. Yeah, you could start now.
Good afternoon to all the respected guests who have joined with us today. On behalf of National Institute of Biomedical Genomics, I would like to welcome you all, especially today's speaker, Professor Ghaiti Hassan, on the eve of the 9th Obaid Siddiqi Memorial Oration. Professor Obaid Siddiqi is one of the few non-US citizens inducted as a fellow of the prestigious scientific academy, that is the National Academy of Sciences US, which is considered the highest international scientific recognition of their contribution to science. Professor Siddiqi was one of the most transformative biologists to come from South Asia and is widely regarded as the father of modern biology in India. He was invited by Homi Bhabha in 1962, the director of Tata Institute of Fundamental Research, to develop a modern biology department, which later became well known as the Molecular Biology Unit. I shall stop here as the founder of this institute and National Science Chair Professor Parthopratim Mojumdar will enlighten us with more such contributions made by Professor Siddiqi and his connection with this institute. With a strong hope that 2022 will be the beginning of the end to this pandemic, we will begin today's event with the invocation song, and the unmasked faces reveal that it had been recorded much earlier.
Sarve स्वस्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां पूर्णं भवतु सर्वेषां पूर्णं भवतु सर्वेषां मङ्गलं भवतु सर्वेषां मङ्गलं भवतु ॐ सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् ॐ शान्ति शान्ति शान्तिः
Now, I would like to invite our director, Professor Soumitra Das, to convey the opening remarks.
Namaskar. I think it's a great pleasure to all of you to welcome and for this event that we annually observe every year to remember our beloved Professor Obaid Siddiqi. As you heard, that Professor Obaid was actually the father of modern biology. And with his inspiration and with the vision of our beloved Professor Partha Majumdar and the then Secretary, Professor M.K. Bhan, this institute was established in 2009. Soon we are going to get into a teenage. So we had this young age problem and the teething problems, I think, in the initial years. And from there, the growth phase has started. Institute has moved into the new campus with the vision to enhance the knowledge of the human health and disease through genomics. As now all of you are aware, the importance of genomics, the importance of the sequence and to know about the mutations and variations. If you really go back and trace the history, it's actually Obaid who pointed out the importance of the mutations long back. And we are actually realizing those in a hard way though, but actually it's really becoming something for everybody's knowledge today. You know, it's a great pride that at the time when India, and not only the India, worldwide, we are going through a very, very difficult time and struggling to really fight back against this notorious virus. NIBMG has actually stood out to the promise. And this is the institute, even in all this 1.5 years of pandemic, the lockdown, and all this difficult time, didn't really close down even for a day. And we are actually proud to say that our sequencing team worked day and night, even now. I mean, I just heard from Arindam, our sequencing team is not even closing their lights. Whole day and night they are working just to cope up with the pace of the sequencing to find out the information and help the nation to actually take informed policy decisions. So, you know, this institute, the mandate, as you know, that we have been working greatly in 4 different areas, just for the people who are actually logged in for the first time and first time hearing for NIBMG. And I'm actually also happy to tell you that today only we have a new addition in our NIBMG family. One assistant professor, Pongali Raghavendra, has joined. He has come from Bangalore. It's so unfortunate that he ended up, but he's now in the guest house because he's not even allowed to get into the main building because of the pandemic situation. But you know, all I'm just so welcome, Raghavendra. So all I was just trying to say that this is the time that we are actually for those people, just to aware you that some of you have heard about NIBMG in the local news channel yesterday, that how this NIBMG is contributing to the sequencing endeavor. But not only the sequence, and in the terms of biology, we are working on the cancer, different cancer. We are working on chronic disease. We are working on infectious disease. And the central to all is the computational biology, where they integrate this information and make it really meaningful contribution. So we have been working on different genomes, and I'm sure Parthuda will also highlight. We are working on the nuclear genome, metagenome. We are working on the mitochondrial genome, and last 1.5 years we have been working on viral genome. So in every sphere, NIBMG is trying really hard to make a dent, and actually they are able to succeed in that. And we are, in this pandemic time, in a different ways, we have contributed and helped the nation to really cope up this struggling time. So, coming back to today's oration lecture, I'm very thankful to Professor Gayatri Hassan. In a short notice, she has agreed to participate. And at the same time, we are sad that we are just not getting her here in the campus. I was just, before this meeting started, I was discussing with her. It would have been nice to have her in the campus, and the sprawling green, you would have enjoyed the Calcutta weather, but certainly not at this time. Maybe in the next time you can just come and visit us and interact with our faculty, students, and the staff. And we are all looking forward for your lecture. And I would request Professor Parthuprati Majumdar, our beloved Parthuda, to introduce the speaker and also give the background of this Obaid Siddiqi Memorial Oration, because that's our small way of actually remembering him and giving our respect to him every year. Over to you, Parthuda.
Thank you. I'm struggling to get my screen up. Vikram, how do I do that? Do I?
I'm coming, Path.
Yeah, please do. Even the. না আমি তো বেরোতেই পারছি না তো আমার এটা দেখানো I'm not able to get out of this. Orinam, are you able to see the screen?
Yeah.
Okay. Yeah, thank you. Next slide. Next slide. Yeah. Thank you. Well, we've assembled here today to pay our tribute to Obaid Siddiqi, who was very close to this institute, and I will have a few things to say about his relationship with this institute as I go along. Let me introduce Professor Obaid Siddiqi to those of you who may not have heard, previous orations. Professor Siddiqui was born on the 7th of January 1932, which is what we are celebrating today. Today is the 7th of January. He passed away on 26th of July 2013 under some very strange circumstances that I will indicate but not describe in gory detail. Obaid Siddiqui got his PhD from the University of Glasgow for his work on microbial genetics with Pontecorvo. He worked across several different model organisms from bacteria to Aspergillus and Drosophila. He studied and characterized the structure of genes in Aspergillus, and later, along with Alan Garan at the Cold Spring Harbor Laboratory and University of Pennsylvania, he discovered suppressors of nonsense mutations in bacteria, which in turn led to the discovery of nonsense codons and the mechanism of chain termination during protein synthesis. So in some ways, he taught us how to read the genetic code and protein synthesis, understand protein synthesis. He also worked in the field of neurogenetics, and in the field of neurogenetics, Dr. Siddiqui along with Seymour Benzer, discovered temperature-sensitive paralytic mutants in Drosophila and the generation and transmission of neural signals. He also studied the sense of smell and taste in Drosophila, including how olfactory information is encoded in the brain of the fly and how chemosensory genetics can be used to study this further. He set up the molecular biology unit at the Tata Institute of Fundamental Research in Mumbai in 1962, and rightly so, he's been acclaimed as the father of modern biology in India. He was a national research professor. He was the founder director of the Tata Institute of Fundamental Research, National Center for Biological Sciences, and we have our speaker today from NCBS. He initiated molecular biology research in India, and that's the reason why he's known as the father of modern biology or molecular biology in India. He, of course, was very well decorated. He was a fellow of the National Academy and, as a matter of fact, all science academies of India. He was a fellow of the Third World Academy of Sciences, an associate of the US National Academy of Sciences, as Anindita has pointed out. He was a fellow of the Royal Society. He also received the highest civilian honor, Padma Bhushan. I want to say one or two little things about his initiation or his connection to this institute. But before that, let me spend 30 seconds about my relationship with Professor Siddiqi. This was a meeting on human evolution that took place in 1997. I was invited to this meeting to give a talk. I entered Cold Spring Harbor, and one of the first persons that I see is Obaid Siddiqi. So I was very pleased to meet with him, and as a matter of fact, I actually got to know Obaid Siddiqi well during this meeting. This was a 5-day meeting, and for those of you who may have visited Cold Spring Harbor, it's really like a hostel where you get to meet everybody, and it's a small place. That's where I got to know Obaid Siddiqi well. I also realized that he was not a speaker in this meeting because obviously he did not work on human evolution or with humans. He sat there from the beginning till the end, every day these 5 days. And essentially he told me that he came to learn about human evolution. And this is 1997, he was born in 1932. So you can imagine what his age was when he was still trying to learn about human evolution. He was very closely associated with this institute. He delivered our first Foundation Day lecture, and the lecture was titled The Development of Genetics. The keynote address at that time was given by Eric Green, who also delivered the 7th Obaid Siddiqi Memorial Lecture. So he was well connected to this institute. I have also known him through other ways. He was the chair of the first task force on human genetics constituted by the Department of Biotechnology. So he was the chairman, I was a member. So I have had very close interactions with him, and I was very pleased when he actually gave our first Foundation Day lecture of this institute. It was under some very strange circumstances that he passed away. The circumstance was that he was walking down the street from his house, and he was hit by a moped. A young guy, his neighbor, essentially hit him with the moped, and that was a road accident. And then he was admitted to the hospital, but succumbed to the injuries. And that was extremely sad for all of us to hear, and for people in Bangalore, including Gayati, to witness what had happened. And so we, when he passed away, and it was subsequent into 2013, the next year we started to celebrate his birthday, and essentially to pay our tribute to him. And today we have the 9th celebration or memorial oration celebrating his birthday. And today, of course, it's Gayati Hassan who's going to give the memorial oration. The first memorial oration was given by K. Vijay Raghavan, who is currently the principal scientific advisor to the government of India. And as you can see, the list comprises a whole host of people, many of whom were Obaid Siddiqui students or collaborators, and they all together actually set up NCBS when it was set up in Bangalore. It was a part of TIFR and was set up in Bangalore. So this is what Professor Obaid Siddiqui, contributed to science, contributed to the organization of modern biology in India. And it's really our great pleasure to have Professor Geti Hassan, who has also been associated with Professor Obaid Siddiqui, to deliver the ninth memorial oration. Her talk is titled "The Genetics of Spinocerebellar Ataxias. Does dysregulated intracellular calcium signaling hold the key?" I will take a pause here and essentially then introduce our speaker today. And our speaker today is, as I said, Professor Geti Hassan, who was quite connected with Professor Obaid Siddiqui. Professor Geti Hassan herself obtained her education in Aligarh, Miranda House, Jawaharlal Nehru University, and the University of Cambridge. Her major scientific contribution has been to obtain a deep understanding of the consequences of changes in intracellular levels of calcium at both cellular and systemic levels. She's been particularly interested in the role of inositol 1,4,5 triphosphate, known as INSP3, and its receptor. For many years, she's been working on understanding the molecular mechanisms of insect flight. Insect flight is very interesting, as all of us know. When the insect takes off on a flight, there is and goes through the flight, there is a lot of bilateral coordination that takes place, and how this happens. And obviously, there are neurons that are involved. So there's a lot of neuron activation and synchronization that takes place. And essentially, Geti spent a large number of years of her academic career trying to understand these kinds of activation of neurons, and during the initiation, the first phase, and later during the flight. Interestingly, while there is bilateral coordination when flight takes place, during courtship, and this is all in Drosophila, during courtship of Drosophila, there is unilateral wing extension. Now, that's very different from bilateral synchronization, and Geti has also studied that, and she's discovered that such asymmetric functions are carried out by independent neural circuits, independent of those that are activated during flight. For her deep contributions to science, Professor Hassan has been elected to all of the science academies of India. She's also received the Wellcome Trust and the Rockefeller Foundation fellowships. She's a distinguished fellow of the Science and Engineering Research Board and has served as a senior professor of the National Center for Biological Sciences. Geti has been a voice that speaks up for upholding the rights of women and minorities in education and society. I have personally known her for decades, and I appreciate her multidimensional contributions, not only to science, but also to social thought. We are delighted that she has kindly accepted our invitation to deliver the 9th Obaid Siddiqui Memorial Oration. She was associated with Obaid Siddiqui for many years, as I just mentioned to you. She will speak to us on a heterogeneous group of human disorders called spinocerebellar ataxias. The title of her oration is "The Genetics of Spinocerebellar Ataxias. Does dysregulated intracellular calcium signaling hold the key?" Geti, the podium is yours.
Okay, thank you, Partha. Thank you so much for that very, very kind introduction. And thank you, Shaumitro, for inviting me to give this talk. I'm, of course, delighted. I hope my screen is visible.
It's visible.
Yes, ma'am. Make it full screen.
Yeah, you
now
can
it's okay.
full screen now?
Yeah, full screen is okay.
Okay, wonderful. Thank you. So yes, it is indeed a great pleasure to be able to talk at this Obaid Siddiqui Memorial Oration. As Partho said, Obaid was sort of a larger than life figure in many of our lives, and I got to know Obaid as professionally as when I joined, actually as a postdoc in Tata Institute of Fundamental Research. sometime in the 1980s, 1984. And that was my first introduction to actually working with genetics. So I had my PhD was essentially in molecular biology, and I was very keen when I started my career that I should be able to use molecular biology in combination with genetics. And I visited India during my PhD, met Obaid, and he convinced me that the way to go about this was to start working in Drosophila. I had never worked with Drosophila before that. And so he convinced me that it was important, Drosophila was the key to doing some of this work. And basically, I decided then, after finishing my PhD, to join his unit in Bombay and start working on Drosophila genetics with molecular biology, of course. And that was a very interesting years, 6 years of a postdoc and being a research associate there, working in combination with Obaid and with Veronica at that time. My longer association with Obaid is, of course, from the time when he was the founding director of NCBS. So while we were working in TFR, Obaid, who's shown here, you can recognize him, is shown here with some of the earlier faculty members who started NCBS. And this is actually a picture from when NCBS was still in the IISc campus. And in fact, I remember meeting Shaumitro around that time. So it's a very, very old picture. And you can see all the various people, including, as Partho mentioned, Vijay, who was our next director, and then some of the other founding members of the NCBS. This is Matthew, Panikar, Sudhir, and Manjari, who then subsequently left the institute. Jayant, unfortunately, is not here. He was another person who was there at this early years. So my subsequent I started my own lab, and I started it in an area which was a little bit different from what I had been doing as a postdoc with Obaid. But then in the process of doing that work, we came together again, and this was a very nice collaboration that we did in the sort of 2002 to 2008, in which both Veronica, who's sitting here, and Obaid, we worked with Obaid's student, Tuhin, and Veronica's student, Pinky, and we worked together for a very nice piece of work on olfaction. So this was probably the only joint paper that I published with Obaid, and it was quite a pleasure. And as I think Partho pointed out, that his intellectual drive was so strong that at this time, you can see at the back this electrophysiology setup. This electrophysiology setup was used by him for the first time, by his lab actually, for the first time, to record from single neurons which are present on the antenna of Drosophila, these flies. And these are these antennae drawn here, and you can see this beautiful map of the antenna and these single hairs, and the recordings were made from this. And this was the reason that we collaborated with him, because we needed this information very urgently for this paper, and so we worked with his student to do this. This is just to give an idea of what an active intellectual individual he was, that right till, this is 2008, and of course he continued till much later. So I'll move on now from this to actually talking about what I'm planning to talk about today. And that is spinocerebellar ataxias. And I hope I can try and explain to you how, from working with Drosophila flight, we reached the field of ataxias. So it's a long story, and but I'm going to try and make it as colloquial as possible and as easily understood as possible. And I hope that if I don't convey something, that people will be happy to ask me questions. So this is a patient who suffers from ataxia, and as you can see, she is not able to coordinate her movements very well without help. And the interesting things about these ataxias is that these are genetic disorders. So that was another reason that we got interested in them. And these genetic disorders are caused by a number of genes, actually a rather large number of genes. So the genes are almost something like 40 different genes cause these disorders. And this is a review that came out on ataxias about 2 years ago, trying to sort of understand, because these genes seem to affect many, many different types of proteins. And they range from something like this small molecule here, which is called the FGF14, which is a growth factor, to the these polyQ expansion repeats, which is found in SCA1 and SCA2 in genes which people don't fully understand what their function is, to many other genes such as this one, which is the metabotropic glutamate receptor. So in this review, what people tried to put together is the idea that perhaps these SCAs all affect different types of channels. And this is what is shown here. So these are different types of channels present on neurons. And the idea is that many of these SCAs possibly affect these channels. And that is what is the underlying theme behind that. What I would like to try and convey at the end of this talk is that perhaps it's not just channels, but perhaps it could even be this kind of calcium signaling which is shown here, and that is affecting these channels, and that is one of the major underlying causes that gets dysregulated in multiple ataxias. And if that is indeed the case, then it should be possible to actually modulate this calcium signaling to try and find ways by which we can improve ataxic conditions. At this point, I would like to mention that ataxias is something for which there is still no cure. So even though they appear somewhat mild in what I showed you this film, but in fact, they do lead finally to somewhat very poor condition of lives. People end up not being able to eat easily, not being able to breathe easily, and it does lead to a sort of early death in many people. Usually they start late around 40 and then cause a lot of problems subsequently, and it's called a progressive neurodegenerative disease. What I'd like to point out to you is this gene here, ITPR1. So this is the IP3 receptor type 1, which is present in humans. And as you can see in humans, it causes these two kinds of, they are both the same gene disease actually, SCA15, SCA16. And in addition, they cause another ataxia, which for some reason is not mentioned here, called SCA29. And then yet another disease called Gillespie syndrome, which is more than just ataxia. It also causes some kind of blindness and other problems. So the IP3 receptor is one of the genes that seems to affect this ataxias. And it has a very close linkage with calcium, which I'll explain to you as we go along in this talk. So, before I move to the IP3 receptor, I thought I'll just try and explain to you why is it IP3 mediated calcium signaling different. So what we do know about calcium signaling in neurons are the canonical mechanisms, which is that you have action potentials that come down the axon and will then cause voltage-gated calcium channels to open. These channels will then bring calcium in, and this is now entry at a very fast scale. So this happens at milliseconds scale. And this calcium then allows for neurotransmitter release, which then acts on the postsynaptic membrane and further neurotransmission. This is what we understand by canonical mechanisms of calcium signaling in neurons. However, as I mentioned, we have other mechanisms of calcium signaling also known, which were actually originally discovered primarily in non-excitable cells. So not in neurons, but in other kinds of, in fact, a lot of them were discovered in immune cells. And this mechanism involves G protein coupled receptors, which binds a ligand, which then act on this enzyme called phospholipase C to activate it, which acts on a membrane lipid, which then creates IP3 and DAG. This is a membrane lipid here. IP3, which is soluble in the cytosol, will bind to the IP3 receptor. And as a result, this IP3 receptor now is not a membrane, it's not a plasma membrane receptor. It's a receptor which is present inside the cell. It's present on ER membranes. Now, the ER is a very strong, it contains a large amount of calcium in it. It's a store of calcium. And when IP3 binds the IP3 receptor, this ligand-gated channel opens and releases calcium. As a result of this calcium release, several activities happen in the cytosol, which are somewhat understood in non-excitable cells. The question that we were interested in asking was that when this calcium is released, what does it do in neurons? does it behave any differently from the calcium that we're talking about here? So for one thing, we know that the dynamics of calcium release can cause very different kinds of changes. And the dynamics of this calcium release is very different from this calcium entry here. This, as I said, happens at millisecond scales. This happens much slower. It happens over tens of seconds. And interestingly, this release of calcium now leads to yet another mechanism, which is called store-operated calcium entry. And these are intricately linked. So what happens here is IP3-mediated calcium release takes place. The store empties out, the ER store empties out, and this needs to be refilled for maintaining calcium homeostasis in neurons. Now, this calcium homeostasis, to maintain this, there is a molecule called STIM, which is present here, which I've shown you here. This binds ER calcium. It's bound to ER calcium all the time. It has a motif that allows it to bind to ER calcium. When ER calcium drops, STIM undergoes a change in conformation. And this is what is shown here. So this is a conformational change that happens with STIM. And STIM now binds to a molecule called ORI. ORI is present on the plasma membrane. So this change in conformation allows STIM to talk to ORI, and ORI now brings in a huge burst of calcium into the cell, which is called store-operated calcium entry. So I will be talking today to you about these three proteins in almost interchangeably because they end up doing more or less the same thing in cells. This leads to a small release of calcium, but as a result of this calcium release, you have STIM changing its conformation and then bringing in large amounts of calcium through ORI, which is then pumped back into the ER and refills this store. And changes through in Drosophila, what we have found is that if you have IP3 receptor mutations or you have STIM or ORI mutations, they seem to do very, very similar things. So I will be using these interchangeably and trying to understand essentially what our lab was trying to understand was when you have mutations in either IP3 receptor or STIM or ORI, then what are the changes that happen in neurons? Are they different from the kind of changes that happen when you have canonical calcium entry? Like, are ion channels affected? For example, does the calcium that is released, does it affect these kind of ion channels? Does it affect neurotransmitter release, which is shown here? Or does it impact neuronal health in any way? So all of these questions is what we wanted to address in the context of IP3-mediated calcium release to begin with, and subsequently to try and understand it in the context of ORI and STIM. So to do this, as Partho mentioned, we work on the fruit fly. And why do we do that? The fruit fly has a brain which is considerably smaller than human brains. It has much fewer synapses. And hence, it's obviously a lot more accessible in terms of understanding circuits, which is important in understanding complex function like motor coordination. It is also blessed with having a large number of genetic tools. So people have been working on Drosophila genetics for many, many years now. Obaid being one of the pioneers who started working on Drosophila neurogenetics. And so a large number of tools have been developed. And one of the key tools that I'll be using to describe to you this work are tools that allow single neurons to be marked and to be accessed in Drosophila. So genetic tools are available by which one can actually express genes in specific neurons. Here you can see the GFP protein expressed in specific sets of neurons in the Drosophila brain. But using very similar methods, one can express almost any other gene of interest that one would like to. And this is what the method that we have used for a long time. Okay, so now to get on to IP3 receptor. So one of the things we did very early in my lab was to develop IP3 receptor mutants in Drosophila. And these mutants, what we found, so these are now mutants which are hypomorphs, which is that they have reduced IP3 receptor function. And when you have reduced IP3 receptor function, one of the first things we've noticed was that these flies are unable to fly. They can walk, they can jump, they can climb, but they are unable to fly. And so when you give them an air puff, just like we've given an air puff here to the wild type fly, you can see that this fly is able to fly immediately, but this fly moves its wings, but it's not able to fly. And our interest at that point was to try and understand where this change is happening. So it's possible, as what I'm showing you here is a very abbreviated flight circuit. the flight neurons are present here in the ventral ganglion, and it's possible that the deficit was here, affecting flight muscles, and then subsequently affecting the wings, or it could affect these small molecule, these small organs called the halteres, which also affect flight, or it could be something more complex. In fact, what we found was that the neurons that seem to affect flight, a large number of them lay in the central brain and were therefore involved in coordinating flight from the central brain going down to the motor neurons, which are present here. And we then got interested in trying to figure out what are these central neurons and how do they coordinate flight. So to do this for the IP3 receptor, I will tell you one of our more recent stories and how we set about doing this. So the IP3 receptor is a tetrameric protein. It is present, as shown here, on the ER membrane, and it's present as a tetramer. And what has been shown by a number of people working on the mammalian version of the IP3 receptor is that it has this ligand binding domain, it has a modulatory domain, and a subsequent channel domain. In the ligand binding domain are present these 3 key amino acids which bind IP3, which are required for binding IP3. And if these are mutated, then ligand binding is abolished almost down to 0. It's completely gone. And interestingly, what this group from David Yule's lab showed was that if all 4 IP3 receptor monomers bind IP3, only then can you get calcium release. If only one IP3 receptor is mutated out of these 4, even if one is mutated, you are not able to get calcium release. Essentially suggesting that if you express a mutant form of the IP3 receptor among the normal forms of the IP3 receptor, and if you get sufficient quantities of this kind of molecules made, tetramers made, then you would essentially create a cell in which IP3 receptor would not function. Even if you created IP3, you would not be able to get IP3 mediated calcium release. So this is what one of my students, Anamika, did. And she created this version in Drosophila rather than in the mammalian version, and then expressed it, as I said, in a specific set of neurons in the brain. So this is the Drosophila brain. And she expressed it in this specific set, which is shown here. These are just 2 neurons, and then looked to see what happens to flight. And when she measured flight in these neurons, you can see that flight is infinitely reduced. So here what we're doing is we're measuring flight time rather than what I showed you. The kind of video that I showed you, very similar assays are being used, but we are measuring flight time. And what you can see here is that these fly for much less time by simply expressing this dominant negative version of the IP3 receptor in these 2 cells. Our question then that we asked was that if this is the case, then why does IP3 signaling in these 2 dopaminergic neurons reduce flight bout durations? Because these are central neurons, and where is their connection with flight? It was not immediately obvious. So to understand this, Anamika then tried to understand what is the actual way by which these neurons are activated. And we had earlier done a screen by which we had shown that a number of G protein coupled receptors were involved in flight. So she tested these G protein coupled receptors and found that this particular receptor, which is called the muscarinic acetylcholine receptor, is expressed on these neurons. And through this pathway that I just showed you, it causes intracellular calcium release. And what we expected was that in the dominant negative version, this intracellular calcium release would not happen. So in fact, she measured calcium from these neurons and showed that indeed in the intracellular, when you express the dominant negative, you are not able to see calcium release here. The point then was that if this is what is happening, if these cells are no longer responding to muscarinic acetylcholine receptor, what exactly are they doing in terms of talking to the brain? So this is the region of the brain that they talk to. This is a higher order center in the brain called the mushroom body, which is involved in coordinating a number of sensory stimuli, olfactory stimuli, and essentially using these to then direct behavior in flies. And what Anamika then did was to see whether in this region that I just showed you of the mushroom body, what happens to dopamine release. So we are using a fluorescent sensor for dopamine here. she's essentially stimulating these neurons here and then measuring dopamine release in this region. And what she's showing here is that when you give carbachol, which is shown here, carbachol being the acetylcholine equivalent, you see an increase in fluorescence here. And this is what happens in control flies. But when the same thing is done in the IP3 receptor dominant negative version, dopamine release is blocked. It does not happen. So essentially telling us that IP3 mediated calcium release in these neurons, in these presynaptic neurons here, is functioning to actually prevent dopamine release at the synapse. So what we suspect is that there is a modulation happening here. That dopamine is acting on these through this medium here, through these dopaminergic neurons, and that this voltage gated channels are also working, and that together this modulation is what is leading to the optimal dopamine release, which then acts on these lower channels here, and this is what leads to loss of flight. Now we have some data suggesting that this loss of flight is related to actually the hunger state of the fly, and that this is what these dopaminergic neurons are doing. They're coordinating this dopamine and hunger state and then allowing the flies to fly for shorter or longer times. For the sake of time, I won't go into that, but I'll move on to another part of the story, which is important and which is essentially more in the context of neurodegeneration. Because what I've talked to you about so far is essentially an acute function of the IP3 receptor. But in the context of neurodegeneration, what we thought was that probably there are some more longer term changes that are happening. And the question then was that what are these long term changes? So we wondered about this, and one of the things that we know that this long-term changes, essentially, as I said, it could be affecting neurodegeneration. And what we wondered was whether this IP3 mediated calcium release and the store operated calcium entry that follows after that, whether any of this is involved in either gene expression or possibly in mitochondrial health. What I'll talk to you today is about gene expression changes, because this is a story that has come along much further. The mitochondrial health part is still something that is coming along in the lab, and I won't discuss that right now. So why did we think of gene expression? Well, one of the main reasons we thought of gene expression is that many of these ataxias that people have looked at have shown changes in gene expression with time. So in the mouse models, for example, people found changes in gene expression. And in fact, even in human tissue, when people have looked at neurodegenerative changes, they've shown changes in gene expression. Now, that is probably not surprising, because neurodegeneration in itself, the neurons will change and gene expression changes will happen. I think our interest was to try and find if this is a causative effect. Is gene expression something that happens, and as a result of that you start finding changes start getting changes of neurodegenerative changes in the neurons. So we began this work by looking at this molecule here. Now I'll switch a little bit and talk to you about Orait, which is shown here. And this is essentially, again, very similar kind of approach that we took with the IP3 receptor dominant negative. We created an Orait dominant negative transgene. Orait is also a tetrameric protein. And again, if you have even one copy of the monomer mutated, you don't see calcium entry. And using this, we looked at dopaminergic neurons and found that, very similar to what we showed in the IP3 receptor, if you express the dominant negative in dopaminergic neurons, you do not see flight. So in fact, it shows a much stronger effect than even the IP3 receptor dominant negative than what we saw. And it's very similar to the IP3 receptor mutants. So these are the two dominant negatives, and this is the control fly. Okay, so what we then went on to do was to see what is the state of the neurons. And essentially, these neurons which express this dominant negative, even with age, they don't seem to change. So now I'm showing you this a control fly, and this is at about 3 weeks, which is actually for a fly is quite old. And what you can see is that these neurons look quite normal, both in the dominant negative version and in the wild type version. So the expression does not seem to cause any neurodegeneration, at least as far as we can tell at this stage. However, what we did want to see was, are there changes in gene expression? And for that, what Shlesha in the lab did was to actually sort out these neurons. So she did a sorting of these neurons using the GFP and then did an RNA-seq with them. And to our surprise, what we found was a huge change in gene expression. And in particular, what we found was that changes in gene expression affected ion transport. So a lot of these channels, which would be critically involved in maintaining the function of the neuron, and interestingly, also in a large number of transcription factors, which would again be involved in, of course, maintaining the health of the neuron because they would be involved in keeping the right genes functional. And so this was something that we were, quite taken aback by and also found rather interesting, because what it suggested was that changes in IP3 receptor and store operated calcium entry, they would affect gene expression and could actually really, impact neuronal health and neuronal function. So to take this forward in the context of ataxias now, I mean, I've been talking to you so far about Drosophila, and really to try and now understand this in the context of ataxias, we really needed to look at this in the context of mammalian neurons and try and understand whether changes in gene expression impact neuron function over time. So to do this, we decided to look at Purkinje neurons, and this is a human patient with ataxia, and you can see that this region of the cerebellum, which contains the Purkinje neurons, is hugely degenerated as compared to a control at the same age. So Purkinje neurons, this is some very nice work that my student Rishabh did recently in trying to understand from the, the Broad Institute gene expression database, what is the expression of these molecules in Purkinje neurons. So what I'm showing you here is that molecules like STIM1, ORI2, IP3 receptor 1 are expressed at very high levels in Purkinje neurons. And so are actually many other calcium signaling genes, intracellular calcium signaling genes, which I won't go into. I'm just pointing out the ones that are relevant to this talk. So these are expressed really strongly. And so Purkinje neurons are clearly cells that use calcium signaling at a very critical way for their functioning. And this is, of course, also obvious from the fact that they are, that IP3 receptor mutants cause Purkinje neuron degeneration. So here are Purkinje neurons. Now I'm showing you this from the mouse cerebellum because we decided to do this work in mouse. And so these are now Purkinje neurons in the mouse. And these are labeled with a red marker, which allows us to mark Purkinje neurons specifically, a red dye. And the green shows you another protein, that is STIM1, which is present also in these Purkinje neurons. So what Dhanya in the lab then did was to make a knockout of the STIM1 gene in these mouse Purkinje neurons. And this is just a very, it's a preliminary slide just to show you what this looks like. So here are the mouse Purkinje neurons expressing STIM1. And what you can see is at 1 year of age, these neurons have completely lost STIM1 from them. They don't have the protein, but what you can see is they are still present. Okay, so the neurons are still present. They do not undergo degeneration. Now, this was a bit of a disappointment to us because we thought they might degenerate, but unfortunately, they do not seem to degenerate. However, we went ahead and continued working with these mice to try and see if there were other effects that we could see in them. And one of the key effects that we found was that these mice were not able to coordinate themselves very well. So motor coordination was lost in them. So what is shown here is a rotor rod test, and these are two wild type mice. And what I'd like you to appreciate is that the STIM knockout mice are not able to stay on the rotor rod as long as the wild type mice are, telling us that they are not able to coordinate their motor function as well. So to then try and understand what was the, sorry, I'll just move on. To try and understand if any of this was related to gene expression, which is what we had found in Drosophila, we took the Purkinje neurons out from these by hand dissection. Actually, I should say Dhanya took the Purkinje neurons out by hand dissection and then did an RNA-seq with them and looked to see whether there were changes in gene expression. And I think the key point that I'd like to make from this work is that not only did she find changes in gene expression, but the changes in gene expression were age dependent. So in this slide, what I'm showing you are some of the key genes that we found were downregulated at 1 year. This is shown here. So these genes are downregulated at 1 year. But the same genes, when we look at them at about 14 to 17 weeks in the mouse, are not downregulated. So you do find a huge amount of genes which are actually downregulated with age in these Purkinje neurons. And this is some of the ones that I'm showing you here are the voltage gated ion channel genes, because as I mentioned to you, ion channels are important for neuronal function. And then of course synaptic signaling, because these are also important for synaptic function is also important for neuronal function. The whole network of genes that Dhania found is shown here. And you can see that there are really a large number of, there's regulation of neuron projection development, ER calcium homeostasis, there's glutamate receptor signaling, because that is one of the key signaling pathways in Purkinje neurons. And as I said, the key point of this work was that we could show that this gene regulation takes place with age, and it's not something that is present at the early stages of the stem knockout. So what we then wanted to try and understand was, if this is the case, then can modulators of intracellular calcium signaling be used to revert these age-dependent changes? So I will introduce you now to yet another molecule, which are called septins. Septins are molecules which are a component of a cytoskeleton. And these are involved in forming some sort of filaments which are present near the plasma membrane and which are able to hold receptors and several membrane protein molecules in place. We got interested in septins very early on because people had shown, this is Anjana Rao's lab had shown, that septins regulate store-operated calcium entry. And so as a result of the work that we had done in flies and shown that there were, loss of flight in both IP3 receptor mutants and in store-operated calcium entry mutants, we looked to see whether septins modulated flight in Drosophila. And what you can see here are different. So these are now stills rather than a movie. You can see that at 0 seconds, none of the flies are flying. Their wings are all static. At 15 seconds, the IP3 receptor mutants are static. Their wings don't move. So as they are at 30 seconds also. But in these IP3 receptor mutants, if you remove a single copy of the septin 7 gene, you can see that they start flying. So they're flying at 15 seconds, they're flying at 30 seconds. And similarly, if you knock down septin 7 in these genes, in the neurons of these mutants, these IP3 receptor mutants, you can see that they are able to fly. So this is the some quantification of this shown on the right, essentially telling us that septin 7 functions as a negative regulator of IP3 mediated calcium signaling or of store operated calcium entry. Now, this is something that we got very interested in. What we imagined was that somehow septin 7, loss of septin 7 was allowing more calcium to enter through the Orai channel and perhaps also affecting in some way IP3 mediated calcium release, though we don't have any evidence for that, direct evidence for that. So our next idea then, in terms of understanding the mammalian aspects that I talked to you about, was that does septin 7 also regulate calcium entry through Orai in mammalian neurons? And to study this, what we did was we created human neural precursor cells and differentiated them into neurons, which is shown here, and then went on to see whether calcium entry is affected in them if you knock down septin 7. So what we have done here is we've taken these neurons, we've kept them in 0 calcium, and then we have added 20 millimolar calcium. A normal neuron with its normal calcium homeostasis will show a very minor change, if at all, in the levels of calcium within the cell. But in the same neuron, same types of neurons, if you now remove septin 7, which is shown here, then you can see that, or you knock down septin 7, you can see that calcium entry is much increased. And clearly this septin 7 entry is through Orai channels, because if you inhibit the Orai channels, you can see that the entry goes down considerably. So this was, of course, in a dish. And our interest, as I had talked to you earlier, was to really try and understand this in the context of motor behavior. So we then went ahead and made what I've mentioned to you earlier. We had a STIM1 knockout mice in Purkinje neurons, which is shown here. We then made a double mutant. So now we have a STIM1 knockout mouse. And in this STIM1 knockout mouse, we removed either one copy of septin-7. So here is the septin-7 heterozygote. And you can see that septin-7 RNA levels are reduced in this case in the Purkinje neurons. Or we made a double knockout of the septin-7, which is shown here. And you can see here that septin-7 mRNA levels are reduced even further in this case. So this is from Purkinje neurons, and you can see that septin-7 levels are considerably reduced. And also, of course, as we look at STIM RNA is also reduced. So now in these mice which have both STIM knockdown and septin-7 partial knockdown, or STIM knockdown and septin-7 complete knockdown, we then went ahead to look at what is the status of Purkinje neurons in this. So these are the key genotypes that I'm talking about. So the first thing we looked at was gene expression, which is what I had talked about to you earlier. And to our surprise, we found that several genes that we had actually noticed, which were downregulated in the STIM1 mutants, were in fact upregulated when we reduce septin-7. So this is in this heterozygous condition, in the septin-7 het condition, and this is in the double knockout condition. And in fact, in both conditions, you can see that the genes are coming back. The expression is coming back to what looks like normal levels, not distinguishable from the wild type levels. We then went on to see whether there was any change in the circuit formation. So I'll just give you a very brief idea of this. Here are Purkinje neurons, and Purkinje neurons receive inputs from something called the climbing fibers. So these are these inputs which come on the Purkinje neuron dendrites. And these are very important because these come from the deep cerebellar nuclei and are sort of required for the normal firing of Purkinje neurons. So what we then decided was to look at these inputs. We had already seen that these inputs go up hugely in the STIM1 knockout condition. So at 1 year, so these are these green dots that you're seeing here. These are the climbing fiber inputs that reach the Purkinje neuron, Purkinje dendrites. And you can see that they are much increased in the STIM1 knockout at 1 year. And we believe that this is a kind of a compensatory effect that the STIM1 knockout mice are doing to try and improve their functioning. And you can see that this is shown here without the dendrites. Okay, when we looked at this in the septin-7 knockout condition, what you can see is that this particular effect is completely reverted. So in fact, giving us a lot of confidence that these mice in which these gene expression changes have been reverted, that in fact, their circuit properties are probably also getting reverted to some extent. And this is what I'm showing you here. You can see that these punctae that are coming from the climbing fibers are considerably reduced in the double knockout condition. But you know, the final test in all of this was that we needed to see what happens to the motor coordination. So what we have seen here is that in wild type, you have STIM1, normal levels of STIM1, normal level of septin-7, and you have normal store-operated calcium entry and calcium homeostasis. We believe that when STIM1 is knocked down and septin-7 levels are normal, that both calcium homeostasis and store-operated calcium entry are reduced. And this is in part because of negative regulation of calcium entry by septin-7. So when septin-7 is present, it continues to negatively regulate calcium entry, and you do not see any change here. You see the downregulated calcium entry. However, when you reduce septin-7, this negative regulation is removed, and now you sort of get back some level of the intracellular calcium homeostasis that was changed. We have not measured actual store-operated calcium entry. We are working towards trying to do this. But what we do find is that calcium homeostasis is probably restored. And what I've shown you so far is the gene expression and the synaptic connectivity is come back to normal. However, as I said, what we were really interested in knowing was what happens to motor function. So what I'm showing you here are the motor function. This is the time spent on the rotor rod, which I showed you earlier. And what you can see here is at 17 weeks, this is the STIM1 knockout mouse, and it spends considerably less time on the rotor rod after being trained. as compared to wild type mice, which are shown here. The septin-7 heterozygous mice are shown here in green. And what you can see is that these are not, I mean, to us, at least at this age, they were not convincingly any different. They could either be part of this group or they could be part of this group. They really didn't look very convincing at all. But with age, we were amazed to find that these mice actually became close to normal. So they went back to the, so essentially what we could see was that the loss of septin-7 could actually revert these STIM1 mice back to showing normal motor coordination. And for this, I'll just show you this movie, which I think is somewhat more convincing, which shows you the wild type mouse. It shows you the STIM1 knockout mouse here, which both of which fall off. And then this is the septin-7 het mice present in the STIM1 knockout, and the septin-7 knockout mouse present in the STIM1 knockout condition. And as you can see, both of these are now able to remain on the rotor rod for longer times. I should emphasize that the time that they remain on the rotor rod is still somewhat reduced as compared to the wild type, but it is definitely significantly longer than the STIM1 knockouts. So just to summarize then, I have reached almost the end of my talk here. What I'd like to just say is that so STIM1 knockdown and septin-7 normal leads to reduction of calcium homeostasis, gene expression, an increase in this climbing fiber PN synapses, and of course a loss in motor learning. With loss of septin-7, we get restoration of calcium homeostasis, we get restoration of gene expression to an extent, we get normal climbing fiber and Purkinje neuron synapses, and we get significant improvement in motor learning. So we think this work is of great, very good, sort of emphasis in terms of spinocerebellar ataxia, because what it suggests is that it may be possible to revert neurodegeneration of Purkinje neurons in ataxic syndromes by modulating intracellular calcium signaling. And in this context, there are now a large number of, compounds being derived across the world, which are involved in actually modulating ORI signaling, which are also in modulating septin-7 signaling. So it's possible that those kind of compounds might come in useful for trying to understand what happens, whether it's possible to improve, the ataxic condition. We think that neurodegeneration is a consequence of calcium-regulated age-dependent gene expression changes. And in this context, I think we are interested in trying to see whether the identified genes that we find which are downregulated, which we of course have not had time to test as yet, whether they can also serve as therapeutic targets for neurodegeneration. So with this, I think I would like to end the talk. I hope I've been able to convince you that from going from Drosophila genetics to spinocerebellar ataxia is actually not such a fast stretch, and that it is actually possible to use a simple organism like Drosophila to try and answer difficult questions like ataxia in humans. I'd just like to take a few minutes now to thank you, to thank the people in my lab who've done this work. So the work on IP3 receptor dominant negative, which I began work with, was done by Anamika, who's shown here. She's now in the final year of her PhD, just writing up and actually just, getting ready to go for a postdoc. The work that was done with Uri dominant negative was done by Trayambak here, and this is his wedding that they're attending. Trayambak married another student of mine, Steffi, and so the two of them are shown here. And Trayambak is now a postdoc in actually a store-operated calcium entry lab, ORI lab, and he now works on ORI signaling in cancer. Then the gene expression work that we started in Drosophila was done by Shlesha, and Shlesha is now a postdoc in Michael Rosbach's lab. So in fact, that work was started in collaboration with Michael Rosbach, and so I particularly wanted to mention Shlesha here. And Shlesha and this work was carried on by Rishabh Mitra, who is now continuing this work and actually working more on trying to understand what brings about these gene expression changes, the kinds of transcription factors that are involved, which I didn't talk about today. Bipon did the work on septin-7 that I talked to you about, most of it, and all the work in mouse, including the STIM1 work and all the septin-7 work, was done by Dhanya, who is also in the process of writing up her thesis and is about to start her postdoc in another couple of months. The human stem cell work, I should mention, Pragya and Ranjita, both of them are shown here. They are the key people who've begun this work in the lab and are actually continuing some of this work in the context of many other degenerative diseases. I would like to mention at this point that one of the SCAs that we have got very interested in is SCA 12, which is, of course, I think there is a link with some of the people in NIBMG with that. And it is something that we are very interested in trying to follow up. And I have some ideas on how that SCA12 might also link up with intracellular calcium signaling. And we are hoping to collaborate with labs in Calcutta and perhaps start this work soon. So hopefully at some point I can talk to all of you about that. I'll stop sharing the screen now, and I'm very happy to take questions if required. Now let me stop sharing. Okay. I hope I didn't go too much over time.
No. Absolutely.
It's fine? Okay, good. Yeah.
Thank you very much, Katie. That was wonderful. Traditionally, in orations, we don't really have any question answers.
Okay, sure.
I don't know whether we want to break tradition, but perhaps we can engage with you on another occasion and ask some questions that we have.
No problem.
Yeah, I saw Professor Vijay Raghavan had joined, and I wonder whether he is still here. If he is still here, may I request him to provide a few messages, especially for our students and junior faculty on this particular day, but I'm not very sure whether he's still here.
Yeah, I'm
yeah, hi Vijay, I can see you. Yeah, do you have any messages for our young faculty and students on this day?
Thank you for springing this surprise for me. I have no idea that I was supposed to say anything. I just wanted to hear Geti's talk, which I did, and it's a really wonderful talk and absolutely, stellar to see how work which started off looking at calcium signaling and the IP3 receptor in a very generic foundational way has resulted in moving through work on mouse and human into extraordinary insight into how calcium can affect gene expression and motor control in important contexts. So that's really terrific. So congrats to Geti and her team for that. Well, what else is there to say? These are very difficult times, and if there's one thing which keeps us together, it's quality science of the kind we heard. And, the analogy is to other difficult times in human history where despite huge wars or plagues or natural disasters, people have, continued with their science in addition to dealing with the context around them. And we should be inspired by those people. I remember, Tomonaga and Gelman, who got the Nobel Prize in Physics for quantum electrodynamics, and the work done by Tomonaga was in Tokyo, which was completely bombed out after the war, and so he was sitting there in the rubble and doing his work, and this is really extraordinary work. This is not a war of that kind, but it's a continuing pandemic which has devastated the lives of so many people. Many have lost their lives, and we should remember them, and we should think about them, and we should do everything to address the pandemic, and at the same time do our science also, so that science and art and music and culture bring soul to our activities and togetherness, which are so important. And attending to these important crises should not mean that we lose our other interactions. So I'm very grateful for this lecture being continued to be held. In a way, the virtual nature of the lecture has allowed many others to come in and join who otherwise may not have been physically present in Calcutta. So that's a very good thing. And Obaid, of course, has always been a tremendous inspiration, both in terms of getting things done in good times, but also being a force for courage and calm in difficult times. So, it's always good to remember him. Thank you.
Thanks very much, Vijay. As I mentioned in the beginning, Professor Vijay Raghavan delivered our first Obaid Siddiqi memorial oration. And today we have the 9th, and we had a series of Still Our People give these orations. Thank you, Vijay, for coming in and saying a few words. Thank you very much. I leave the podium to Anindita, who will conduct the rest of the session.
Thank you, Vijay.
So we would like to thank Professor Gaiti Hassan for apprising us with her significant contributions towards intracellular calcium signaling, both in Drosophila as well as in mammals. And we also want to thank Professor Vijay Raghavan for joining with us today. So, as we know, Professor Obaid Siddiqi was very fond of string instruments. We would now like to switch over to a short cultural event and start it with a prayer song to the Supreme to bestow peace in these times of anxiety and despair sweeping all over the world.
So let's start the cultural program.
Yep.
very relevant song of Rabindranath Tagore, which says, Amar Mukti Aloe Aloe. Amar Mukti Aloe Aloe, Ei Akashe Amar Mukti Aloe Aloe, Amar Mukti ধুলায় ধুলায় ঘাসে ঘাসে এই আকাশে আমার মুক্তি আলোয় আলোয় দেহ মনে সুদূর পারে হারিয়ে ফেলি আপনারে দেহ মনের সুদূর পারে হারিয়ে ফেলি আপনারে গানের সুরে আমার মুক্তি ঊর্ধ্বে ভাসে এই আকাশে আমার মুক্তি আলোয় আলোয় আমার মুক্তি সর্বজনে মনের মাঝে দুঃখ বিনোদ তুচ্ছরা কঠিন কাজে আমার মুক্তি সর্বজনে মনের মাঝে দুঃখ বিনোদ তুচ্ছরা কঠিন কাজে বিশ্বধাতার যজ্ঞশালা আত্মের বন্ধি জ্বালা বিশ্বধাতার যজ্ঞশালা আত্মের বন্ধি জ্বালা জীবন জানো নিয়াহুতি মুক্তি আসে এই আকাশে আমার মুক্তি আলোয় আলোয় এই আকাশে আমার মুক্তি আলোয় আলোয়। Life is an enigma. Why do we all are facing this conundrum? Here is Soumya Chatterjee with another relevant song.
We would now like to invite our Associate Director, Professor Orinder Moitro, to conclude today's event through deliverance of the vote of
Thank you, Anindita. So, in these difficult and challenging times of the pandemic, life must go on and find new ways of going about it. So, we remember Professor Obaid Siddiqi, his memories of his first footfall in our institute during its very early years after birth. And these memories will remain etched in the soul of the institute as long as it will go on. We take this opportunity to thank sincerely and convey our gratitude to the honorable speaker in today's oration, Professor Geti Hassan, for taking time out of from a valuable and busy schedule and illuminating us with this wonderful talk. Thank you, ma'am. We remember our engagement with you in the days when you were members of our scientific advisory committee, and we hope to continue to engage with you in future.
Thank you. Thank you. Yeah.
So we thank Professor Vijay Raghavan, sir, for taking his time out and joining this event, which transformed today's event to a completely new dimension. Thank you, sir. We look forward to your footfall in the new campus in more favorable times.
Thank you. That exaggeration notwithstanding.
So now we thank our director, Professor Soumitra Das, for his continuous support and guidance and ensuring that we are able to organize this event and his enthusiasm in getting us to organize this event. Thank you, sir. I would like to thank now Professor Parthapratim Majumdar, but then the predicament is for n number of things I thank him for, there are too many things which remains unmentioned. So Professor Majumdar has been the one who took the initiative in founding this oration. The first speaker was Professor Vijay Raghavan, and throughout till this last 9 years, every oration of this event, he has ensured that he provides his unstinted guidance and support and help in for us to organize this particular event. Thank you, Patroda, and we continue to look forward to your support in future. So this is the time. Thank you, Patroda. This is the difficult times, and we appreciate that the many distinguished scientists and luminaries and the rest of the audience who came and who joined us online in this event and connected with us. We thank you for your solidarity and your joining us in this event, and in future we look forward to more favorable situations where you will be able to visit our institute if you have not done so already. Last but not the least, I would like to thank the members of the team which organized this event behind the scene. The entire members of NIBMG community and family who all put their solidarity on the event and ensure that in difficult times where nothing can be taken for granted, not even the status of health of individuals which were changing in matter of hours, not days, that this event could be organized. In particular, I would like to mention my colleagues, Professor Anupam Basu, Dr. Maulina Tacharya, Dr. Soubik Mukherjee, then again Dr. Alpana Dave, and the Sri Rajiv Kumar and Mr. Sri Nabarun Chatterjee and the entire team of administration. In particular, Sri Bikram Rai and his IT team, which is instrumental in getting these kind of online events done, and the rest of the community for their support. Thank you very much. At the end of we have come to the end of this today's event, and though for those of us who are present in the campus, I invite you on behalf of the entire institute to join us for some small packed snacks available at the entry of our main building. And for the rest, we look forward to meeting you in the campus in better times. I wish you all a very enjoyable evening and a great and safe 2022. Okay. Goodbye.
Thank you. Thank you, Arindam. Yeah.